Yeah, hello everyone. I would like to welcome you to the QIAGEN webinar series, New Tech, New Discoveries, so the Human Microbiome Research webinar series. And our talk today is presented by Seth Bordenstein, Why Diversity Matters in the Human Microbiome Science. My name is Christian. I'm Global Product Manager at QIAGEN for basically all microbiome related kits, which you can purchase at QIAGEN, and I will be your host for today's session. So let me introduce you to our speaker for today. So Dr. Seth Bordenstein, he is the director of the One Health Microbiome Center and professor of biology and entomology at the Penn State University. So Dr. Bordenstein is a really internationally recognized microbiome scientist, which I think also leads to the huge attendee number already, which we have here in our webinar. And he is in the science area over 25 years. And his research is uncovering how microorganisms are shaping life, shaping health, and also the environment. And I'd say his work spans mosquito-borne disease control, human microbiome diversity, even into microbial trends across the animal kingdom. So he's also leading one of the largest microbiome research centers in the world with over 500 members, if I'm correct. And he's working at the intersection of agriculture, environment, and health. And Dr. Bordenstein is also the founder of the global science education program, Discover the Microbes Within. Amongst his many honors are awards for excellence in teaching and research and endowed chair in microbiome science and recognition as a very highly cited researcher. So I'm super happy to have him here. But before we are finally welcoming Dr. Bordenstein and begin the presentation, we have a few housekeeping items which I would like to go over. Today's session will be in listen-only mode. So we encourage you to submit any questions you may have during the presentation through the Q&A box. And we will try our best to answer your questions during the session, but also at the end we will have some time left. There's also, like always, a legal disclaimer before we begin. We need to note that when there is any QIAGEN product shown or mentioned here, the intended use is molecular biology application, and these products are not intended for the diagnosis or prevention or treatment of the disease. With this, I will now hand over the presentation to Seth. Seth, are you ready to take over?
Ready. Thank you very much, Christian. Appreciate the introduction. And I'd like to say good morning to all the listeners. Thank you for joining from what looks like around the globe. That's pretty exciting to see. I am joining you from the middle of Pennsylvania at the tip of what we call the Appalachian Mountains in a very rich green forestry mountain area. And Penn State University is sort of right in the middle of all that. It's a large university with 50,000 undergraduate students and a very large faculty to accompany that undergraduate pool. I want to go ahead and talk to you today about a topic that we've been thinking about for about 10 years, and it's really motivated by a big question, which are, what are the rules of microbiome variation, both between species and within species? And that latter part is very interesting for the talk today because, you know, we focus on a lot of our attention on the headlines of what defines the human microbiome sciences. But when we sort of look a little deeper, we tend to find things that are a bit more nuanced than maybe what we read in the headlines. And so what are the rules of microbiome variation within humans is the topic today. Now, just in case there are some new beginners to the microbiome field joining us today, I wanted to just introduce why we are also fascinated by the microbial world and a little bit about the One Health Microbiome Center that I direct. So when you look up into the night sky, it's often we have this feeling of wonder, enchantment, using a telescope about what is up there. Is there life there? What don't we know yet? And for many microbiologists, it is really important that we communicate that that same wonder and enchantment occurs when we look in a microscope. Because when we look in a microscope, we can see tiny dots that make us wonder about what's there. How much don't we know yet, and what can we discover? And so in fluorescent shown here are tiny viruses and then tiny bacteria as well. And it's with this backdrop that a lot of us and our colleagues are so excited about the last 20 years in the microbiome sciences as we kind of begin a new field of discovery, looking down instead of looking up. And by and large, that's the motivation for our own center here, which we've termed the One Health Microbiome Center. This is a merger between two life science fields that are growing in prominence over the last couple decades. The first is anchored in the Microbiome Centers Consortium, which is about 80 centers across the world that are joined by a common consortium. This has obviously been an explosive time for microbiome centers across the country and the world. But at the same time, there's about 100 academic units studying One Health, which is defining that the health of one ecosystem is dependent upon the health of another ecosystem. And by bringing these two large fields together and essentially anchoring it with the fact that microbes are the base of the biosphere, the One Health Microbiome Center understands and recognizes that human health and its microbes are going to be determined by the flow of microbes from the environment and agriculture, and in all directions that health can be impacted. And with that, we have a lot to say about the way the field's going to move forward, because we believe that it can be unified and unsiloed in a way that it maybe hasn't had a formal experience just yet. In that regard, our center is well represented with 550 members from students to faculty, 125 faculty in 42 departments. We have partners and collaborations in 100 countries. We offer the first microbiome sciences PhD degree in the world, and I imagine that this upcoming submission cycle, there might be some graduate students or undergrads interested in going to grad school in the microbiome sciences. Please let them know that they can get accredited with a degree here as a first in kind, so that's front and center on their resumes and CVs when they graduate. We also work closely with a number of projects. One of them is the Discover the Microbes Within the Wolbachia Project, which is our flagship biotech science education series, where we offer free microbial-based labs in biotechnology. You're encouraged to check that out if of interest. And then we also work in a partnership with QIAGEN, where we are essentially delving into establishing resources, benchmarking new kits, and doing co-promotion with QIAGEN. So we've had a wonderful time working with the QIAGEN group. Now, at our center, which reflects this kind of broader push to define One Health and put it into action, including by the National Academies of Sciences. One way to think about it is that, every model system, every non-model system, of course, has a microbial community. And those microbial communities are reflected in these circle charts where the bars represent different types of microbes. And of course, the flow of those microbes could shape the composition of those colors or of those microbes in those ecosystems. Within our center, this is just a small picking of faculty who are represented in each of these ecosystems as microbiome scientists. So for example, Jordan Bassange is using artificial intelligence and chronic diseases to predict what types of microbiomes could be engineered to offer resilience in the gut microbiome ecosystem against C. difficile and other types of inflammatory bowel diseases. Estelle Cordeau is a soil microbiome scientist trying to understand erosion, nutrient cycles, and the health of soil through a microbial lens. And Dr. Erica Gonda, who studies all types of livestock microbiomes, is in the agricultural realm. And she's very interested in chicken microbiomes, cow microbiomes, and how they can be optimized with probiotics or different types of feed. And the list could go on, but this gives you a flavor of how we are bringing together lots of different microbiome scientists from different ecosystems under one large tent. Now, the talk today is anchored in the human microbiome field, and it's often sort of appreciated that the microbiome of humans is quite diverse. And that's clearly the case. There are about 50% of the human cells in the body are human, but the other 50% of the cells in the body are microbes. And so this provides an enormous appreciation for the fact that our bodies are not just human, but they are microbial, and gives a perspective of holobiont biology that we definitely appreciate. The other sort of fun fact to disseminate is that if you think about what are the number of microbes in the mouth, it turns out that there are more microbes in the oral cavity than there are a number of people on the planet. So we have a whole world of microbes inside each of our mouths. And some of our scientists are looking at how to optimize the oral microbiome to prevent cavities or to cure cavities. For example, Laura Wyrick is an example of that, where she's developed the first oral microbiome transplant for curing cavities. In a broader context, the body is an ecosystem with different parts. So every part has its own microbiome that's usually distinguishable in some form. While there can be overlap in microbes between different sites, different body parts tend to have different compositions. There's an estimated 40 trillion microorganisms in and on the human body. There's an estimated 100 times more genes in the microbiome than the human genome itself, which offers an enormous capacity for functions across the body. Of course, we know that our own genomes across the world are very similar, and also that our microbiomes can be very different, that we can often be 80% different in our microbiomes between any two people at the species level. And so one of the big questions for the human microbiome field is, while our genomes are so similar, why are our microbiomes so different? And what are the common factors and forces that are shaping that variation? Both variation and if there's similarity between us. 90% of diseases have a microbiome association. So of course, the relevance of understanding the microbiome variation may be directly related to understanding why we have predispositions and differences in chronic diseases that affiliate with microbiome variation. Now, if we were to ask sort of in a textbook, you open the textbook on the human microbiome and ask, well, what are the major rules that have defined the microbiome sciences in humans so far? And these are a few of them. On the first few pages, you'd be reading that chapter, and there's clearly geographic variation in microbiomes across the world. And humans, of course, are affiliated with those environmental microbes. So human variation in microbiomes correlates continentally across the globe. But even within our body sites, as we were mentioning, across our body sites is a biogeography of different microbes across the human anatomy. Now, in westernized countries, there's also a general trend towards microbiome diversity declines, that as westernization, industrialization, modernization has taken place, microbiomes tend to lose a significant fraction of their diversity in terms of the number of species present. And on the same hand, there's an increase in the rise of chronic illnesses in those same areas, which has led to the hypothesis that perhaps these diversity declines are linked to the rise in the chronic illnesses. And so with that backdrop, we're sort of left with a lot to think about. What still makes microbiomes different and what makes them similar? Now, human health is profoundly impacted by microbiomes across the board. If just looking at the gut microbiome, for example, there are all kinds of literature links to a number of different diseases. And I'm sure one of you in the audience has a connection to one of these particular diseases, or everyone in the audience has a connection to one of these diseases. They're just so common. And that begs the question of what is driving that variation across individuals in these chronic illnesses, and what types of microbiomes might they be correlated with and causal by. So bringing us back to this particular perspective that the human microbiome is a wild ecosystem composed of half of our cells are microbial, and they are highly abundant. Now, when we look at literature reports, actually, before I get into that, I want to remind some of you in the audience that from our center, I mentioned earlier, Jordan Bassans is working on engineering the gut microbiome to prevent C. difficile. So he's used artificial intelligence and mouse models to now actually whittle down to a single bacteria that can prevent C. difficile colonization and illness. Not just 20 bacteria, not just cocktails of bacteria, but a single bacteria that could, in theory, be used as a human probiotic. And as I mentioned earlier, Laura Wyrick is looking at oral cavities and has found that she can design a microbiome for transplant therapy into the oral microbiome and effectively cure severe cavities with a 100% efficacy rate. So just two examples about why we need to understand variation in chronic illness and how microbiomes can help. Now, the rules of microbiome variation within humans is sending a strong signal to our field, because this is a study that was done by some of my colleagues based out of the University of Chicago now in Ron Bleckman's lab, where they looked at the number of human clinical cohort studies and found that most microbiome studies are concentrated in a single background, European white ancestry. And over time, there's starting to be an appreciation that we have skewed a lot of our microbiome analyses to one particular ancestry and region. And so you can start to see chipping away now at increasing amounts of diversity over time. But overall, 70 to 90% of human clinical cohort studies have been done in this white European North American background. And of course, we've made the same mistake in many ways that genomics has, where genomics went too fast and concentrated in one particular population, leaving behind diversity. And so it's kind of surprising that the microbiome sciences, did exactly the same thing, and we left diversity behind. And that's starting to be appreciated. In the United States, there are two publicly available data sets that really founded the field, the American Gut Project and the NIH Human Microbiome Project, both of which began in the early 2000s and really defined the field. And so I'm going to concentrate our studies today on diversity in American data sets that are publicly available. But of course, the questions that we talk about are profoundly important for across the world. And in those data sets, we are using numbers that are reflected here from the American Gut and the Human Microbiome Projects. These are the number of subjects we used in our studies. And the reason we wanted to do this is to ask essentially, what are the rules of microbiome variation? But when we do that with respect to diversity, because we need to unbiased microbiome studies. That's clearly the case from the charts I just shown. We need to think about health disparities, not just health across all of us, but the health in particular groups that are afflicted from different rates and incidences of diseases. And we know that many of the ones that are differentially preferential across ethnicity and race are ones that are correlated with microbiome variation, including colorectal cancer, obesity, heart disease, etc. Now, when we think about health across diversity, we need to think about a lot of factors that are occurring at once in individuals and in populations. Many things can vary across populations and individuals, including access to healthcare, frequency of medication, socioeconomic status, the neighborhoods we grow up in, and the environmental exposures we're placed to, and our biology and genetics. And so with that, it's going to be a very high level talk about thinking about diversity at very high levels, but understanding that underneath diversity is a whole set of complex factors that can define our own microbiomes and the populations that carry them. In this talk, we'll use the terms race and ethnicity. In fact, these are required terms in all NIH clinical cohort studies. And they are social constructs, not biological constructs, and they correlate with determinants of health that have biological consequences. And so when you think about a gut microbiome that might be dominated by Firmicutes and Bacteroidetes, the two very large abundant gut phyla, these phyla can change in abundance with respect to disease, so type 2 diabetes or colon cancer, but they also could change with respect to self-identity and social identity. So in this case, if there are different microbiomes in the gut for colon cancer between two different groups, that might have something of importance because it might also link with the relatively difference in increases in the incidence and mortality from colon cancer across these self-identity groups. And so it's important that we baseline sort of what is microbiome variation across ethnicity and self-identity so we can appreciate potentially their influence to these disease difference incidences. Health disparities are understood as preventable differences, things that we can get ahead of, such as access to healthcare or environmental exposures. And so if we can also eventually get to a point where we can offer guidance for microbiome maintenance and getting ahead of these disease incidence differences through the microbiome, that could potentially be of use as well. So ultimately, the goal is to get towards a place of health equity by understanding microbiome variation across these self-identity and social groups. Now, self-reported identity, such as ethnicity and race, are large social constructs. They've often been referred to as ghost variables because they hide a lot of the detail underneath the hood, as we were just talking about. And so it's important to remember that these are very complex social terms with different forces affecting each individual and populations. With that understanding, I want to show you some of our initial forays into comparing the American Gut Project data and the NIH Human Microbiome Project data, because we wanted to filter out sort of what is a common trend across data sets versus what isn't. And when you look at adult 16S microbiome diversity at the phylum level, you can kind of see broad differences and slight differences in the ratios of Firmicutes and Bacteroidetes across these 4 groups that were included in the 2 studies. If you dig a little deeper there and ask, well, what are the headline variables that link in with this human microbiome variation across these 2 data sets, you find something pretty interesting, which is there are 3 factors that are significant across only one of these studies, HMP or American Gut: age, sex, and BMI. Only one of those is significant in these 2 studies. The only factor that was significant in both studies was the self-reported identity, ethnicity, and race. And that factor explained more of the variation between humans than any of these other factors. So there was 5.1% of the variation was explained by these. Now, in the human microbiome field, that's actually a pretty good number. Human genetics is thought to influence about 1 to 10% of human microbiome variation, and it's difficult to find very large factors. So I just want to put that percentage of the variation explained in context. But the most important thing being that this is a consistent factor as opposed to these other ones that sometimes get a lot of attention. All right, so if we go a little further down and we can start to look at specific types of bacteria that vary between self-reported identity groups, we can find 12 reoccurring genera or families in the gut that are consistently variable in the same exact direction, meaning more abundant in one group versus another, for these bacteria. And interestingly, 11 out of the 12 of these bacteria have what have been deemed heritable in many different studies. Heritable meaning that genetic variation in the human genome can influence the microbiome variation in the gut, so the abundance of these microbes. And oftentimes, these microbes co-associate with each other. They kind of rise and fall together. And they have been termed the Christensen-Eliaceae consortium in that regard, because this is a key microbe that these microbes are rising or declining with. Christensen-Eliaceae is known as the most heritable microbial member of the gut. About more variation in the abundance of this microbe is explained by genetic variation in the human genome than any other microbe in the data set. Now, Christensen-Eliaceae has been studied by Ruth Lay's group and many other labs to be associated with obesity. And so this chart just reflects that, but it's drawn across these 4 different population groups now. So when the color is not filled in, Christensen-Eliaceae is absent. And then when the color is filled in for each of these groups, Christensen-Eliaceae is present. As you can see in 3 out of the 4 groups, when Christensen-Eliaceae is absent, tend to have a higher BMI than when it's present. And that has been known from previous studies, including in mouse models. But here we can kind of see a granularity that not all groups show this correlation. And when certain groups do show this correlation, there's differences in the magnitude of those. It makes us think harder about looking at Christensen-Eliaceae in biology across these particular populations with a little bit of a finer comb. Now, one other tool we can use to emphasize that microbiomes do have repeatable differences is AI and machine learning models for microbiome pattern recognition. And the basic premise here is that if we feed lots of microbiomes into the machines and they look for feature recognition that distinguish particular individuals or groups, it would be able to identify a certain individual or group. And in this case, it might have a high confidence in identifying my own microbiome if we had trained the machines well. When we look at machine learning model data, it's often plotted on a false positive against true positive rate. And the perfect classifier will have a correlation that looks like this. Okay, and a random classifier will have a line, a diagonal line straight through the middle that looks like this. Okay, so when the true positive rate is highest, you're going to have this box type plot, and when the true positive rate is essentially equal to the false positive rate, you're going to have this diagonal line. Now, we worked with Ron Bleckman's group between our lab and his lab, and ultimately did this for the American Gut Project data and the Human Microbiome Project data across the 4 groups. And what you can see here is that there's pretty good, I'd say moderate levels of predictability of these distinguishable microbiomes between the groups. And that leads to a % accuracy. The models can generally predict self-reported ethnicity or race with a 70 to 80% predictability. So it's not perfect, but it's definitely above the bar of a random classifier. So emphasizing that, again, what we see in statistical analyses can also be shown with machine learning models. Now, that all was done in adults. Liz Malott was a former postdoc in my lab. She's now at WashU in St. Louis. She did this on children and found that the distinguishability was even higher in children than adults. So these models now are trending towards a very strong classification for children age 0 to 12. And she found ultimately that microbiomes could be predicted with 90% accuracy. So that's much higher than the 60, 70, 80% accuracy in adults. It turns out that if you take the distinguishable microbes from adults and you take the distinguishable microbes from children, interestingly, 50% of these taxa overlap between children and adults. They're not always in the same direction, but at least the taxa themselves are predictably important to this distinguishability from children to adulthood, suggesting that maybe what happens in childhood also lingers on in adulthood. And so you can see that overlap here. Okay, and I think a lot of you have probably, if you know the microbiome field, have heard that human microbiomes change with age. They start off with relatively low diversity, meaning low amounts of different types of taxa, and then that increases over time to about 3 years of age, where microbiome diversity tends to plateau at that point. So the childhood age is an influential age that could set up longer term patterns in our gut microbiomes. And that's the data shown here that Liz pulled together from a number of different studies on childhood microbiomes. Now, if we look at the same type of analysis that we did for the adult microbiomes and look for a correlation with self-reported identity, prior to 3 months of age, there's no link. There's no association. And that's intrigued us because it suggests that what happens early in life is not establishing a distinguishable microbiome. And I should qualify that. It's what's early in life prior to 3 months, because what happens after 3 months starts to get more interesting. This is when we start to see a link with gut microbiome variation emerge now. And from 3 months on, so this is 3 to 12 months, if you go all the way up to 12 years of age, you can continue to see this association with self-reported identity persist. And so this was kind of interesting to us because it suggested that perhaps life exposures and experiences during the early months of childhood are then setting up these microbiome differences that could persist in the longer term. Now, we don't know what those early life exposures are. We didn't have that kind of granularity to the metadata, but it does suggest that something about the lived experiencing is setting up these microbiome differences. Okay, so when we have conversations around this, one of the things I like to talk about with my colleagues is, what is the factor that's shaping this microbiome variation that's consistent across so many different studies? And generally speaking, we tend to focus in on diet as a lived experience factor that's influencing gut microbiome variation. And so we set out to test that. And we did that in adults who were from Nashville and who are aged 18 to 40 years, who are all female, so we're controlling for any potential influence of sex, who all had a healthy BMI, and who all previously ate a conventional Western diet. And they were distinguished between 2 self-reported identity groups. And in this study, which was a relatively small study, we had 36 healthy females across a total of 2 cohorts, we did a dietary intervention to essentially ask the question, if you're on the Western diet and then you are exposed to and eat a full vegetarian diet, will that cause any convergence in the microbiome variation? And then after they go off the vegetarian diet, what happens after that point? Do they sort of diverge again? And so the basic structure of the experiment is give everybody the same exact diet, which was quite expensive, and that's why we only had a small cohort size in this pilot study. But if there is a dietary effect, we should see that microbiome differences in the beginning of the study converge after being on the same diet, and then they might reemerge when they come off the same diet. Alternatively, if diet has no effect, we should see these microbiome differences persist because there's some kind of legacy effect or some other non-diet factor that's influencing the differences. So here is the dietary intervention. It's 3 plant-based meals and 1 snack per day. Everybody's eating the same food, but the calorie content is personalized to maintain weight. So slight differences in the amount of calories to maintain their incoming weight. And unfortunately, everybody had to suffer through not having any coffee or caffeine, no chocolate, and no alcohol. So all the vices that people rely on were eliminated, but in part these were eliminated because they can have high metabolite profiles, and we did measure metabolites in the study, and they can largely mask other microbiome-mediated changes. So things like caffeine, if you cheated and had a coffee in this study, we would have known that and seen a caffeine spike in the metabolites. And then I think there was one person that it did experience that, and we cut them out of the analysis. Okay, so the measurements we did were across the body: saliva, blood, urine, stool. Now, I'm not going to be able to talk about all these today, and the multi-omics spanned metagenomics, metabolomics, and viromics. This is an ordination plot or a graphical plot of the habitual diet shown in this large gray circle for cohort 1 and cohort 2. And then the study diet, the intervention where we controlled the vegetarian diet, are shown in the brown circles here. And so for cohorts 1 and 2, there are significant differences between the habitual diet and the study diet. That's important because it shows you we're changing the microbiomes with the dietary intervention. There's a bit more difference in cohort 1 and cohort 2, but both are statistically distinguishable between the study and the habitual diet. Now, looking at their microbial communities across the study in both the fecal stool microbiome and the saliva microbiome, what you can see is that each individual here is color-coded. So one individual's in pink, one individual's in gray, one individual's in green for both the fecal microbiome and here the oral microbiome. As you can tell, the individual's microbiomes are largely held tight throughout the study, that an individual holds on to their microbiome even though they've been going through a dietary intervention. So there appears to be a legacy effect of what you start with can only change modestly with what you end up with if the dietary intervention is to have an effect on the microbiomes. This is showing very much the same data, but in a different form, that the dietary intervention really has a negligible effect on the gut microbiomes. This ordination plot shows the before, during, and after phases of the gut microbiomes in cohort 1 and cohort 2, and there's really very little difference, although it's statistically significant. These values are so modest that there's almost no change in the microbiomes. Now, the gut microbiome variation at the strain level also persists across dietary stages, and this is now gut microbiome variation between the 2 social identity groups. And as you can see in cohort 1 and in cohort 2, there's persistent differences in all the stages, which are just combined here. And that is a fact that continues throughout the study. And because we did metagenomics, we not only needed to look at strain level, but we could also look at metagenomic functional categories in the gut and oral microbiome communities. And those categories are shown as COGs, which are conserved orthologs in the gut microbiome, ARGs, or antibiotic resistance gene profiles, and then carbohydrate digestive enzymes, or CASIMs. And we looked at this across the gut metagenome, the oral metagenome, and then the gut virome. And as you can see, there's a number of structural differences in these functional categories that persist across the study. COGs are different across all these sites that we sampled. ARGs, or antibiotic resistance genes, are different across all the body sites that we sampled, and CASIMs are as well. And CASIMs showed a massive difference where 33% of the variation could be linked to the self-identity group and the oral metagenomes. And interestingly, there was a very large difference in Cazymes for fiber digestion versus sugar digestion, suggesting that maybe past dietary preferences might have influenced an enrichment of fiber versus sugar digestion in these Cazymes. So it's pretty clear the message here that it's difficult to converge the microbiome with a dietary intervention because there's such persistence at the strain level and at the metagenomic level across this dietary intervention. Now, to kind of bring it together, there's a lot of data that we accumulated, but this is a summary heat map of essentially what was differential across cohort 1 and cohort 2 and across the various functional categories we measured, and then across self-declared ethnicity or race, the use of antibiotics. Because females were our subjects, we also analyzed hormonal contraceptive use, and we did that for each one of the microbiome sites sampled. And here's how I want you to focus your attention, that in cohort 1 and cohort 2, the gut microbiomes at the taxonomy level are significantly correlated with ethnicity, antibiotic use, and hormonal contraceptive use. So that is a persistent factor that is quite strong. The other factor that is of interest is that the gut virome is even more statistically different than the gut microbiome across taxonomy, across COGs, conserved orthologs, and across antibiotic resistance genes. And so this, to me, actually told me something new in the study, that the virome is in some ways a more sensitive biomarker of variation at multiple levels across these two cohorts of the study. And in thinking about this, the gut virome makes a lot of sense to be more distinguishable. In an early study in 2010 by Reyes et al., they actually showed metagenomic functional categories that are color-coded here for the gut virome and then for the gut microbiome. And as you can see, the gut microbiomes of these 10 individuals or so were color-coded, and the metagenomic functional categories are identical. But the gut viromes are like a wild ecosystem. They're incredibly diverse. And that makes sense, that the gut virome is highly variable, both from evolutionary forces and ecological forces, and are offering a lot more variation for the statistics in our study to detect than the gut microbiome, although clearly taxonomy is still an important factor for the gut microbiome. The trend line for the use of ethnicity and race in the microbiome literature has increased over time. This is a great article that I referenced in the beginning by DeWolf and M. Systems. And it's pretty clear that this is not sustainable because these terms are ultimately mysterious terms that don't define what the causal factors are underneath the hood of these terms. So what we're beginning with now, with an angle towards understanding baseline microbiome variation, we'll need to move to another phase where this use drops down and we start to get more granular about the various lived experiences that define why microbiomes are variable. And it's in that theme that I just want to end on where we began with a little bit of, we need to unbias microbiome studies across human development from age, young age to old age. If we're going to have an influence on health disparities, we really need to bring in other kinds of metadata factors. And ultimately, if we're going to be successful, we'll learn enough to develop ways to have inclusive therapeutics, inclusive diagnostics, inclusive guidance and policies for microbiome management and awareness. The participation of social scientists in the microbiome sciences is, I hope you've gotten a sense that this is imperative, that it'll change the dialogue and discoveries, because biologists can't do this on our own. The science is moving fast, and it's not just about social effects, it's environmental effects and geography and pollution, and population health scientists are well poised for this type of work. What do we need? We need longitudinal data across the lifespan, which is obviously challenging from an organizational funding and financing perspective. We need functional mechanistic studies of whose people contacts are, real high level or low granular level data on diet, stress factors, etc. We need better experimental population designs, not just comparing between groups, but really going deep on within self-identity group analyses. We need social intervention strategies and policies eventually, if we gain enough knowledge to curb microbiome disparity links. and ultimately develop more personalized therapeutics to engineer and alter the microbiome. A one microbe fits all model is great, but how realistic that is may be challenged for some of the diseases that have disease incidence differences and where the microbiome differences are also apparent as well. Now, in thinking about the whole system, the holobiont, if you will, of the human host and its microbes, Rob Markowitz was a former graduate student and has moved on into the industry world, asked this question. Do microbiome-linked human genes associate with diseases across biobanks? And I want to put this in light of self-identity groups as well. And I'll show you that in a second. So in the database, we analyzed known human microbiome-linked genes from 90,000 subjects at the BioVU database at Vanderbilt. And these were stratified by European and African ancestry. And there were 900 collated genes that Rob pulled together from the literature that were scanned in this biobank and then studied for disease microbe associations. And intriguingly, what we found and what Rob found is that there were 31 associations of human microbiome-linked genes across 10 genes in the genome for European ancestry across various types of diseases shown on the x-axis here. And then their location across the chromosomes are shown here. We're not going to get too granular with this, except that we could not find any association with individuals from African ancestry. And to me, this also speaks to the fact that either we need to raise our awareness and increase sample sizes of understudied groups, which is definitely a factor in the African ancestry group, or we've got to take a more holistic look and thinking about disease incidence differences and having different holobiont level differences in terms of their genetics and their microbiomes. And this is a big thing going forward for the field. There's been lots of discussion on self-identity and the use of ethnicity and race in the human microbiome field, and there are really good articles out there talking about this. And on the same hand, we need to think about what can social scientists who very much are comfortable in this space help us out with, and think about understanding human microbiome variation from a more lived experience level. And that lived experience, of course, begins early in childhood, and our data is suggesting that there is a time period after 3 months of age where microbiome differences start to emerge. And we wonder, over time, as the microbiome develops and the immune system develops and as we age, is that setting up something more longer term as a legacy effect on our adult microbiomes and therefore diseases that accumulate later in life? And finally, I'll just end with some high level thinking on what is a host biologically made of. And, we tend to think about things from the nuclear genome perspective, which is composed of all sorts of helpful and also parasitic genes like meiotic drive elements and transposons. And for the most part, the nuclear genome is vertically transmitted, but gets shuffled through recombination. And, if we integrate this more holobiont level thinking and put the microbiome into the host as what makes it up biologically, I think we end up with a more complete picture. And as the talk today illuminated, our genomes can be very similar, but our microbiomes can be very different, and they can tell us hidden stories in both the bacterial world and the viral world. Of course, there are all sorts of helpful and harmful and harmless microbes. And of course, there's a predominance of environmental or horizontal acquisition in the microbiome, but some of them can be vertically transmitted as well. So there's a lot of conceptual thinking that can bind the nuclear genome dynamics with the microbiome dynamics. And in total, the whole system can be more fully understood from a phenotypic perspective by bringing all this information together. So with that, I'd like to end on prioritizing diversity in the human microbiome sciences has been a goal for us over the last decade and continues to be. We've found trends that begin at 3 months of age in the United States and then develop and continue into adulthood. We found that on our dietary intervention, there was no dietary effect. Happy to discuss why that may be or hear your thoughts. And that the virome, it really opened our eyes to holding prominence in what is more sensitive to detecting than the gut bacterial part of the microbiome. And taking this holistic genetics, nuclear genetics and microbiome view, I think will be important to ultimately understanding traits of interest that we're so curious and want to make an impact with. Finally, I'll just end with a note for y'all that at the One Health Microbiome Center, we're celebrating World Microbiome Day on Friday, June 27th. And Curtis Huttenauer, who's a human microbiome pioneer and established a lot of the tools of the Human Microbiome Project and works in various diseases, we'll be giving a lecture, an open public lecture by Zoom, and the QR link, if you just hold your camera up and get that link, you can register through it, and I encourage you all to join us there. We're looking forward to hanging out with Curtis for an hour and talking science with him. With that, I just want to say thanks to everybody for listening. I noticed there were some questions coming in the chat box, and I assume those will be moderated for about 10 minutes or so. This is just a small picture of the One Health Microbiome Center. We love to do microbiome science across ag, environment, and humans. And if you're looking for a place to hang out with or collaborate or join in center-level global projects, please think of us as you consider that. And with that, I am happy to take questions.
All right. Thank you, Seth, for this really great presentation. Was a pleasure to listen to it. And
my pleasure.
It's great that we have a certain period of time now for the questions, because we have been busy during your presentation. So there have been a lot of— I
could see the chat box going.
A lot of questions, and I tried to also summarize some of these questions, because, as you can imagine, a lot of them were going in the same direction. So yeah, let's start with it. So a lot of questions were going into the direction of these two cohort study you have done, where you introduced the diet. One is challenging, or a lot of them are challenging the timelines which you have applied. So thanks a lot for this insightful talk. I wonder if the few days of intervention are sufficient to cause a significant change in the profile of gut bacteria. Or I wonder, however, if it's possible to draw strong conclusions about the impact of diet over such a short period of time. Ecosystems take time to change, and allowing only a few days may not be sufficient to draw conclusions, and so on. So these are the questions going into this direction.
Yes, and I really like those questions, and as everybody might imagine, we've thought about those as well. And I think, you know, one of the caveats, of course, is that it's a 4-day dietary intervention, in part limited by how much money we could spend, because giving everybody the same meal ends up turning into hundreds of thousands of dollars a year. So that design, however, is not totally crazy. There are several studies in the literature that suggested even within 24 hours, you can flip a microbiome that was once vegan to carnivore, or vice versa. So we actually started this experiment based on that precedent and thinking 4 days would be sufficient. Obviously, having the data in hand now and not seeing that difference certainly raises the question, maybe it doesn't move in 4 days, maybe it'll move in 4 months. Maybe it'll move in a year. And of course, that would require a major amount of resources and investment. And that kind of study isn't sustainable under really any funding mechanism right now. While we can, you know, add a component in across months and years, fiber, for example, we can do small nutrient changes, but you can't, you know, it's real difficult to fund a whole study controlling the diet. It also made us think about what the literature actually says about diet. And the more we looked at diet and its influence on the microbiome variation, the more we actually found after the study that there's quite a lot of variability in whether the diet can change a microbiome and how long of time it takes to change it. And so diet does get a lot of headlines, and I think rightfully so, but it's also not clear that it's universally influencing microbiomes in a way that maybe hasn't gotten enough attention. And so we have, I think, a lot to learn still. And obviously a lot to think about in terms of, is a 4-day dietary intervention telling us enough? Yes, it is. I mean, it's telling us that at least under that period of time, we couldn't see a change. And of course, the trajectory from childhood to adulthood suggests that these differences might be long as well. Could we fund a longer term study? That would be great. So if you know anybody, we'd love to do that.
Okay, now you hear me, right?
Yes.
So another question area is going into probiotics. one question was, have you tried probiotic-rich diet to see changes in gut microbiome? And the other is going a little bit into a pathogen-related area. You mentioned that Clostridium difficile growth can be prevented by one single bacterial species. How does that happen? And do they have C. difficile tailored toxins or other ways to specifically prevent colonization by C. difficile via these probiotics? So these two questions, a little bit going in different directions.
So the diet we showed was all vegetarian based. It wasn't sort of enriched with fiber. It really was a switch in whole nutrient profiles, as you can tell, which we thought actually was rigging the system in some ways to change the microbiome, but that only had a modest effect. And on the latter question, the C. difficile single bacteria that prevents colonization resistance, I'll refer you to a paper by Jordan Bassans in Cell Hosted Microbe that came out this year. Probably just type those terms in Bassans, Cell Hosted Microbe, you'll find it. It turns out that it's a nutrient utilization pathway that allows this particular probiotic to essentially prevent C. difficile colonization as they utilize and compete for similar nutrients. And so the details can be found in the paper, but also, I'd be happy to make connections for folks that want to look into that work a little bit further with Jordan Bassans.
Okay. And then we have already a couple of questions which are going into the direction of the diet and birth related microbiome of children. So the one is babies born via a C-section tend to have a different gut microbiome composition compared to those born vaginally, but tend to converge by the time the child reaches toddlerhood. In your studies, you showed that gut microbiome variation persists throughout childhood, suggesting also a long-term impact on health. Did you include the mode of delivery, C-section, vaginal birth, into your studies, as there might be a different lasting impact on the gut microbiome composition of children? And the other is going into the direction, since microbiome not fixed at birth, has there been a comparison of microbiome virome and children raised vegetarian versus not? And yeah, but that is a similar question, vegetarian, vegan, omnivore. So
those are great questions. And clearly, we're scratching at the surface of, I think, a relatively, new topic for us to think about in the human microbiome sciences. So the good news is that there's a lot of work to do, and I hope that people see that and want to do it too. The study, I'd have to check the details with the first author on how C-sections and vaginal birth were controlled. It might have been that we had that in statistical analysis. But one of the things we did check was, were any of the early life differences in the microbiome taxa related to potential vaginal acquisition to the child, right? So are the same microbes that are differentially abundant after 3 months also linked to vaginally transmitted microbes or in the vaginal canal itself? And we didn't see any evidence for that. Also affirming that the differences appear to be arising not from, a vaginal to childhood experience and not from an immediate birth experience, but from a lived experience that is accumulating over time. And we can see that signal at the early stages of 3 months. We use publicly available data for the early life childhood work. And so most of the data is in the bacterial part of the microbiome. There aren't really a lot of studies on the virome, although they're just beginning in the last few years. So I think at some point we'll be able to do a similar type analysis on the virome part of the early life childhood. Thanks for those really good questions.
Yeah, and we have so many more. So I need to also think about which ones are most relevant for this audience here.
Yeah, right.
I think one person had a really interesting question in regards to heterogeneous genes and microme in relation to therapy success and also the origin of disease and dysbiosis. So dysbiosis is the imbalance of a microbial community or microbiome that lives in the human body, for example. And he's asking, when we talk about microbiome in certain diseases, say, for example, inflammatory bowel disease, why it's still not clear whether dysbiosis in gut is caused or the consequence of the disease. And the other is, my second question is about heterogeneity, I hate this word, of genes within a patient population, how each patient responds differently to a particular therapy. Is it linked to heterogeneity in microbiome within a population?
Right. Those are two very big questions that are difficult to answer right now. Fecal microbiome transplants have had an enormous number of successes for treating C. difficile and more rare other diseases. And by and large, they have not been done with a lens on, that the fact that microbiomes might have differential responses to individuals. And including, the background at which these self-reported identity groups might have. Traditionally, the microbiome's cleared out before a fecal microbiome transplant. So let's say a dose of antibiotics, and then the fecal microbiome transplant occurs by a colonoscopy or something like that. So there's a washout and then a reconstitution of the microbiome. But if it is not sort of reflective of the prior legacy effects of that person or population's microbiome, then you would want to ask that question, sort of like, well, what are the long-term consequences of this? And of course, we don't have data on that. I think we're only going to see data emerging on that in the next 10, 20 years as people who once received a fecal microbiome transplant develop healthy or not. And that's certainly something to keep an eye on. I do think that there's a slight increasing recognition of granularity about what microbiomes we're putting into people. But unfortunately, the data hasn't and the tools haven't developed fast enough for us to really provide recommendations and policy guidance on this. But that's exactly what we'd love for all of us to be discussing based on this type of talk today. And I'm so glad that many of you are thinking about it in this way, because if we don't, then we're ignoring the diversity and we're ignoring the applications and tools that we develop for a one-size-fits-all that might lead us astray. And there are certainly cases where fecal microbiome transplants in rare cases have gone astray. Mm-hmm. So with that, I think that is the most important part of this talk, is making us all think harder about moving with a more fine-tooth comb through our microbiome successes and applications in the future, especially for human health.
Okay. And there are certain questions which are going a little bit beyond. They are going into which techniques you have used, how did you sequence certain things, and if you can give more information regarding oral cavity. I assume that we can forward you these questions and that you are answering at your own will, and also to which extent you would like to.
Definitely, and a number of these papers were published in PLOS Biology. So if you want to also look into the methods there, they can be found open access online.
Okay, for the sake of time, maybe I have two more questions. One is, a great session. What is your take on neurodegenerative disease and diet?
Yeah. Thanks for the question. I'm not an expert in this area, so I'd leave this to my neuroscientist and microbiome colleagues. But from what I'm seeing from the pioneers of this field in gut-brain axis, there's a strong sense that they are considering and doing studies where diet influences the gut microbiome, which in turn could influence mental health in some capacity. Whether it's a chronic illness that's severe, whether it's an anxiety-related trait, those things are up for the grabs and experimentally. And I'm excited about what some of my colleagues are doing in that space. But I would be remiss to sort of say anything too prematurely about any facts that are emerging out of that area. But I'd say stay tuned. It's an important part of, I think, the future of gut-brain axis is taking into account how diet can modulate that.
Okay. And the last question, and I think that is, it's also a big one. It's going into the ethical direction, but maybe it's also
Sure.
a nice closing one. So as far as I understood, the observed microbiome variation is not caused by race or ethnicity per se, but by lifestyle and social determinants of health that often align with these categories. How do you estimate the risk in future that the ethnicity microbiome variations could be declared as purely biological or genetical racial differences? These interpretations could reinforce harmful racial stereotypes, also suggesting that certain groups have healthier or less healthy microbiomes without any context.
Yeah,
it's a big one.
It's a big one, and that's obviously why we are put so much effort into describing, the fact that this is a good discussion going on right now. That indeed the ethnicity and race are social constructs. And so that's why we emphasize so much that this is really a lived experience. But, if we ignore that, right, if we ignore that to the risk of individuals who would otherwise want to turn this science upside down, then we also ignore that to our own peril. And so we shouldn't let them win the conversation. We should have this conversation, obviously speak about it, and move towards a better future with positive impacts for humans. And that's exactly where, this needs to go in order to have that kind of impact. Because, every funding organization in human health recognizes that there are health disparities and that it's an important area to understand more about, especially from the social sciences, who are already very knee deep in this particular area. What hasn't happened, though, is getting the microbiome, which is a socially environmentally acquired aspect of our life. And as we've seen in our own data, that this becomes part of the story. And the merger and education and professionalization of social sciences and microbiome sciences is the answer to making that better future, and not having something twisted in an upside down direction. And so I'm optimistic that we can do that as scientists, and we should do that as scientists for making a better future for everybody.
All right. I think we are coming to an end here for the sake of time. We'll close this first presentation of the whole session. So thank you, Seth, for this really wonderful presentation and the insightful Q&A part. Thank you all for joining today. And any questions that we weren't able to answer during the session will be answered later by email, hopefully by you. We will at least forward it to you, Seth. And now have a great day, everyone. And it was a pleasure. Bye.
Thanks, everybody.